My dear students, welcome to my today's online class on amplified fragment length polymorphism, and by abbreviation it goes like this: AFLP. You have learned RFLP, RAPD in my previous classes. Now we shall learn one more such technique, and at the end try to compare and contrast all the three techniques that have been discussed so far. So today we are discussing about amplified fragment length polymorphism, AFLP. So I am Dr. MEERA B, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and email ID, and after the class any queries you can contact me. Now you can see one more picture, which is the schematic representation of the technique what we are going to learn today. Okay? Yeah, this is the reason why I am highlighting it. AFLP is a very sensitive method for detecting polymorphism in the genome. Same job is done by RFLP, same job is done by RAPD, but of the three, AFLP is quite sensitive method for detecting polymorphism in the genome. It is based on the principle of RFLP and RAPD. In a way, it is a hybrid of RFLP and RAPD. Okay. It is appropriately regarded as diagnostic fingerprinting technique that detects genomic restriction fragments. I have not taught you DNA fingerprinting so far. In my next coming classes, I am going to teach that. Right now, you just remember that AFLP is treated as a diagnostic fingerprinting technique, and what it does, it detects genomic restriction fragments. Here, we make use of PCR amplification rather than Southern blotting for the detection of restriction fragments. By now you know that in RFLP, we learned that for scoring both PCR and Southern blotting are used, but in AFLP, we make use of only PCR amplification and not the Southern blotting. This is a difference between RFLP and AFLP. Very important thing to be noted is AFLP is employed to detect the presence or absence of restriction fragments, please note, and not the lengths of these fragments as done in RFLP. This is the major difference between AFLP and RFLP. I repeat, AFLP is employed to detect the presence or absence of restriction fragments, not the lengths. Okay, so that makes the technique quite sensitive. All right. So since it is very important, I have highlighted this way. AFLP is widely used in plant genetics rather than animal genetics. I'll let you know why it is. See, normally AFLP is not used for mapping animal genomes because this technique is mainly based on the presence of high rates of substitutional variations which are not found in animals. But such substitutional variations resulting in RFLPs are more pronounced in plants. Thus, AFLP finds applicative value more in plant genetics rather than in animal genetics because substitutional variations are normally not found in animals. This is a point to be noted. The basic principle of AFLP is the amplification of subsets of RFLPs using PCR. Alright, let's see what exactly is done. So, in this slide, I am going to take a particular example and explain the technique. So, see this schematic representation. What all is being done, let us see. A genomic DNA is isolated and digested simultaneously with two different restriction endonucleases. You can see here is the genomic DNA. On one side you have the sequence GAATTC, which is the recognition sequence for EcoR1. On the other side you have the recognition sequence TTAA, which is the recognition sequence for the restriction enzyme MSEL. Thus, EcoR1 has a recognition sequence with a 6 base pair, whereas MSEL with 4 base pairs. Now, this is the genomic DNA, and here is one site for EcoR1 and one site for MSEL. Both of them are restriction endonucleases. Now treat this DNA with both the restriction endonucleases simultaneously. Then what happens? These two enzymes will cleave the DNA and result in small DNA fragments which are less than 1 kb so that they can be amplified by PCR. So in the second step you can see you will get the restriction digested products. Alright. Then for this purpose, to run the PCR reaction, the DNA fragments are ligated with EcoR1 and MSEL adapters. Till now, I have not taught you about linkers and adapters concept, which are often used in recombinant DNA technology. See, once you digest the DNA with restriction endonucleases, there comes a concept called modification of cut ends of DNA. There are around 10 methods by which you can modify the cut ends. By now you know that they produce either blunt ends or 5' overhangs or 3' overhangs depending upon the way the restriction endonuclease cleaves the DNA. I have taught all this in my previous lecture. Now, once you get the cut ends like this, you can always modify the cut ends so as to suit to your experiments. There are around 10 methods, and out of 10, one such method is adding the linkers and adapters. Okay. So, just right now you can just know that you will add here EcoR1 adapter and MSEL adapter. Adapters can be designed for different restriction endonucleases, and accordingly you can use whichever adapter you want for your experiment. Okay. So to run the PCR, before running you have to modify the cut ends, for which you have to ligate the DNA fragments with EcoR1 and MSEL adapters. See, here is the case. This side I have shown EcoR1 adapter and this side I have shown MSEL adapter. Right? They can be designed in the laboratory. Then, these common adapter sequences, flanking genome sequences, serve as primer binding sites on restriction fragments. Why do you add adapters? To serve as primer binding sites on the restriction fragments. You know, PCR can't be carried out without the primers being binding to the DNA fragments, right? Then the DNA fragments can be amplified with AFLP primers, each having only one selective molecule. In the initial stage, you will add first primers with one selective nucleotide, okay? One selective nucleotide. And you will get the PCR products. And these PCR products are diluted, and again they are used as templates for selective amplification, employing two new AFLP primers that have 2 to 3 nucleotides. This is the next step, right? Addition of first primers with one selective nucleotide. Once you get PCR product, then addition of second primer with 2 to 3 nucleotides, okay? Then, after the selective amplification by PCR, the DNA products are separated on a gel. I'll show you in the next slide. The resultant DNA fingerprint is identified by autoradiography, and these AFLP fragments represent unique positions in the genomes and hence can be used as landmarks to bridge the gaps between genetic and physical maps of genomes. Okay. In plants, AFLP is useful to generate high density maps and to detect genomic clones. So whatever I have shown in this slide, I'll just repeat. A genomic DNA is isolated and digested simultaneously with two restriction endonucleases, EcoR1 and MSEL. Okay, then the two enzymes will cleave the DNA and result in small fragments less than 1 kb, which are amplified by PCR. To do this, the DNA fragments are ligated with EcoR1 and MSEL adapters. These common adapter sequences serve as primer binding sites on restriction fragments. The DNA fragments can be amplified with AFLP primers, each having only one selective nucleotide first time. Then whatever PCR products you get, they are used as templates for selective amplification. Employing two new AFLP primers that have 2 to 3 selective nucleotides. Okay, that is the next step. Then after the selective amplification by PCR, the DNA products are separated on a gel. The resultant DNA fingerprint is identified by autoradiography, and AFLP fragments represent unique positions in the genomes and hence they can be used as landmarks to bridge the gaps between genetic maps and physical maps of genomes. Okay. So in plants especially, this is useful to generate high density maps and to detect genomic clones. Okay. Let us see in next slide. Same thing, whatever I explained, I have put it here for your reference purpose as well as for reading purpose. This serves as a study material. Okay. Same thing. So 2 to 3 nucleotides, then you will run on a gel by autoradiography. Now, again, this is a very important concept, what I have stressed already, right? They serve as landmarks to bridge the gaps between genetic and physical maps of genomes. Then, you can see this schematic representation. What all I have explained with one particular instance, Same, I have given a general schematic representation. You will isolate total genomic DNA. First step, you will conduct restriction digestion, making use of restriction enzymes. You will get so many DNA fragments. Then you will do adapter ligation by putting the adapters which are designed for particular restriction endonucleases. Then you will do pre-amplification. Then you will do selective amplification, and whatever fragments you get, that you will subject it to gel electrophoresis. And this slide gives you the overall picture of what is done in AFLP. Yeah, this is another schematic representation briefing you about the various steps involved in AFLP. So genomic DNA, you put restriction enzymes for digestion. Then you will give adapters for ligation. Then you will get ligated DNA fragments. You will amplify them, right? Selective primers. And you will get amplified fragments. And you will run on polyacrylamide gel, okay? There are two types of gel electrophoresis: AGE, agarose gel electrophoresis, and PAGE, polyacrylamide gel electrophoresis. In this case, you have to run PAGE. And you will get the bandings like this. Here they have taken 3 samples of DNA, A, B, and C. Look at the fingerprint pattern. You can make out that sample B and C are almost same fingerprint pattern, whereas the pattern obtained for sample A is different, right? This is how AFLP is done. Now this slide briefs about RFLP versus RFPD versus AFLP. In RFLP, it is a codominant marker. RFPD is also a codominant marker. AFLP is a dominant marker. RFLP, non-PCR based approach. RFPD, PCR based approach. AFLP, combination of both RFLP and RFPD. RFLP, there is need for restriction endonucleases. RFPD, no need of REN, whereas in AFLP, there is need of restriction endonucleases. RFLP requires large quantities of DNA. RFPD, very meager amounts of DNA. And AFLP do not require large quantities of DNA. Then RFLP primer is not needed because this is a non-PCR based approach. RFPD, it uses multiple arbitrary primers and lead to unreliable results. That is one drawback of RFPD. Whereas AFLP technique uses only 2 primers like PCR and gives reproducible result, reliable result. Then in RFLP, no such rule to use restriction enzyme. Anything you can use. RFPD, you don't use restriction enzyme at all. AFLP, combination of restriction enzymes are used. Okay, frequent cutters and rare cutters. So this slide briefs you the RFLP versus RFPD versus AFLP. Right? It gives a comparative account of the 3. Then we have to discuss advantages and disadvantages of these 3 methods, whatever you have learnt. Coming to RFLP, advantages: paternary test, it is used for identification of criminals in forensic medicine and to identify genetic diseases. Disadvantages: it is slow, more tedious, quite costly, and requires more DNA samples. On the other hand, AFLP is used, has advantage: positional cloning of gene of interest, high reproducibility, then many loci can be analyzed simultaneously. Disadvantages: complex procedure, non-identical Commigrating bands can cause noise. That is one problem. RAPD, genetic relation establishment, genetic diversity studies, analyze mixed genome samples, applicable when limited DNA is available, works out economical. Disadvantage, markers are dominant, that is, they can't distinguish whether DNA sequence is amplified. DNA sequence amplified is heterozygous or homozygous. So these are the advantages and disadvantages of RFLP, AFLP and RAPD what you have studied. You can see one more marker here, SSR and ISSR. So they are also a type of markers. I have not taught you because this is not there in the syllabus. So dear students, till now I have explained you the AFLP technique and I have given a comparative account of all the three techniques whatever you have learnt, and have also highlighted the advantages and disadvantages of the three techniques. Okay. Thank you for your patient listening. Any questions you do, contact me. Stay blessed. Thank you.